Hello, and welcome to this lesson on the way in which cannabinoids interact with the receptors of the human body. We will be discussing the most common and pharmaceutically relevant cannabinoids. Specifically, our focus is on how they are able to affect both our cannabinoid receptors and other receptors found in the body. Here is a list of many cannabinoids, including both major and minor cannabinoids. This is meant to act as a review of which cannabinoids are commonly seen in cannabis products, and is certainly not exhaustive. While some of these are naturally produced in the plant in significant quantities, others are not. Please take a look at the structures of these molecules, as their structure dictates their interaction. Notice how certain compounds have 3 connected rings, while others have 2 or 1. This can make a huge difference in how they interact with receptors. Now we will move on to an overview of CB1 receptors. CB1 receptors are a type of cannabinoid receptor primarily located in the central nervous system, with high density in the brain and spinal cord. However, CB1 receptors are also found in other organs in lower density, such as the liver and kidneys. CB1 receptors play a role in many physiological processes, including mood, memory, appetite, and pain perception. Overall, evidence points to the purpose of CB1 receptors being to help the body maintain homeostasis, or a state of balance, by regulating certain signaling processes. Within the body, CB1 receptors are activated by our endogenous cannabinoids, which are the cannabinoids produced inside of our body. These primarily consist of 2-arachidonyl glycerol, most known as 2-AG, and arachidonyl ethanolamide, otherwise known as anandamide. However, it is believed that there are other endogenous cannabinoids as well. Cannabis products people consume contain phytocannabinoids, a form of exogenous cannabinoids, which means they are cannabinoids produced outside of the body. Now we can move on to how those exogenous cannabinoids interact with CB1. The following lists the phytocannabinoids by the general strength in which they have an interaction with CB1 receptors. Certain terms used may be unclear. However, I will do my best to clarify. THC is a partial agonist, meaning it activates the CB1 receptor to a certain extent, but not fully. It also has psychotropic properties, or is associated with a feeling of being high. You can think of an agonist as something that activates the receptor. Now, CBN has low affinity for CB1 and acts as a partial agonist, but much weaker than THC. This means it does not interact as strongly with the CB1 receptor as THC does, and its psychoactive effects are only seen in large doses. CBG has low affinity for CB1 and can act as a weak partial agonist. This means it does not interact very strongly with the CB1 receptor and may only partially activate it. CBG is non-psychotropic, meaning it does not produce a feeling of being high. This is in line with the weak interaction, and you will see that the rest listed are also non-psychotropic. CBDV is a very weak partial agonist, meaning it only slightly activates the CB1 receptor and is therefore also non-psychotropic. Moving on, THCV has been shown to be an antagonist or an inverse agonist at CB1, meaning it can hold the space of the receptor and block the activation of it. This may be why THCV has been believed to suppress appetite. However, it may also act as a partial agonist, meaning it can partially activate the CB1 receptor, but it has been suggested to do so in a way that inhibits cyclic AMP formation and stimulates the beta-arrestin 2 protein. CBD acts as both a weak antagonist and a negative allosteric modulator of CB1. This means it can block the activation of the receptor, and it can also bind to a site other than the main receptor dock. This causes a change in the receptor's conformation or shape, and decreases the receptor's affinity for agonists. It is common knowledge that CBD can affect the way THC impacts the body, and modeling suggests that CBD will turn off the receptor just after THC activates it. THCA, the acidic form of THC, has been shown to have a very weak interaction at CB1, and it does exhibit minimal agonist activity. However, it is hard to fully classify due to unavoidable THC contamination. Thus, it does not interact strongly with the CB1 receptor, and its effects may be difficult to distinguish from those of THC. This is not saying that it lacks an effect on its own, but because THCA can turn into THC easily, it is hard to gauge. Now, CBC may be a weak antagonist, and it could also modulate signaling mediated by the CB1 receptors, but the mechanism is unclear. Finally, CBDA has minimal to no bonding or efficacy at CB1. Now it's time to review the CB2 receptors. CB2 receptors are a type of cannabinoid receptor that are primarily found in the immune system, with high density in white blood cells and the spleen. CB2 receptors play a role in a variety of physiological processes, including inflammation and immune function. When activated, CB2 receptors modulate the release of a wide range of molecules throughout the immune system. CB2 receptors are not found in the brain in as high of a concentration as CB1 receptors. As a result, they are not thought to play any role in the psychoactive effects of cannabis. Instead, they are believed to be involved in the body's response to injury or infection. The following slide lists the phytocannabinoids by the general strength in which they have an interaction with CB2 receptors. Being that the CB2 receptor system is not as well researched, this information is more limited in scope than that of CB1 receptors. First, we have THC. THC acts as a partial agonist at CB2. The interaction of THC with the CB2 receptor is similar to its interaction with the CB1 receptor. Now CBN. CBN has been shown to be a partial agonist at the CB2 receptor, similar in strength to THC. CBN is much stronger at CB2 as it is at CB1. Next, we have CBC. CBC has been shown to be a partial agonist at the CB2 receptor. Similarly, CBDV is a compound that acts as a partial agonist at the CB2 receptor, and is much more effective at this receptor than it is at the CB1 receptor. For CBG, there is evidence that CBG has low affinity for the CB2 receptor and acts as a partial agonist at the receptor. Moving on to CBD, it has been thought to be an antagonist at the CB2 receptor. However, recent evidence suggests that it may also be a partial agonist at the CB2 receptor. Overall, it has been thought that CBD may exhibit its anti-inflammatory effect on the body by acting to inversely express CB2 receptors, which in turn inhibits immune cell migration and reduces clinical signs of inflammation. Now THCA. It has extremely slight agonist activity at the CB2 receptor, and again is weaker than CBD. Similarly, as mentioned earlier, it is difficult to classify its activity at this receptor due to the unavoidable presence of THC contamination in many samples. Then we have THCV, which is a compound that acts as an antagonist at the CB2 receptor. However, it may also act as a partial agonist. Finally, there is CBDA, which has minimal to no bonding or efficacy at the CB2 receptor. Now, let's discuss the way in which cannabinoids have the ability to interact with the receptors outside of the endocannabinoid system. Emerging research focusing on cannabinoid interaction with receptors outside of the endocannabinoid system is becoming increasingly popular. While it is likely that many of the cannabinoids mentioned previously have diverse interactions, we will focus on some of the key points to grasp from these 6 cannabinoids shown here. The overarching thing to remember is that the structure of cannabinoids points to how diverse their interactions with other receptors will be. So, cannabinoids with 3 connected rings, such as THC, CBN, and THCV, are less likely to significantly interact with other receptors due to their rigid shape as molecules. For example, THC mainly binds to the orthosteric or primary binding site of cannabinoid receptors, and it does not show significant binding preference outside of the cannabinoid system. Given that CBN and THCV have similar structures, it can be assumed that they would also follow similar interactions. Taking the serotonin receptors as a point of comparison, there isn't evidence of direct interaction from THC or CBN. As for THCV, there is some evidence that it may affect the activation of 5-HT1A receptors, but that this is likely indirect and not through binding in the main site. Being that THCV has the shortened side chain, it has the biggest structural difference out of the 3. Now, as we look to the other 3 cannabinoids, CBD, CBG, and CBC, we see more open structures with either 1 or 2 connected rings. This coincides with greater interactions at other receptors. For the serotonin receptors, CBD has been shown to bind to and act as an agonist at 5-HT1A, which is stronger than its interactions within the endocannabinoid system. However, it does not stop there, as CBD has evidence of affecting the activity of opioid receptors, PPAR receptors, and TRPV1 receptors as well. This is why it is described as a promiscuous molecule, since its structure allows for it to bind to and impact many other receptors. Considering those receptors impact a range of physiologic processes like pain, mood, inflammation, and even gene transcription, it is no surprise that CBD is able to produce a range of physiological effects. Now, although CBG and CBC are also much more flexible and dynamic molecules than the tricyclic cannabinoids, they have very distinct shapes from each other and from CBD, so each should have a diverse set of interactions outside of the cannabinoid receptor system. As of this moment, there is not as much research on these 2 cannabinoids outside of the CB receptors. However, given their structures, it will be no surprise if other interactions are found. Putting it all together, the most important thing to remember is that the structure of a molecule dictates how it interacts with receptors. THC was shown to have the strongest interactions at cannabinoid receptors, and seemingly does not interact significantly outside of that system. CBN, which has the most similar structure, unsurprisingly has the second strongest interactions within cannabinoid receptors. Then, as the shape of the molecule deviates further, more diverse receptor interactions become possible. But it does not necessarily mean that there will be interactions. Thank you for watching. I hope that this helped you to understand a little bit more about what is going on inside our bodies when phytocannabinoids are ingested.